Hi, good morning. I'm Carlos Cuesta. I'm CEO of Oryzon Genomics. Oryzon Genomics is a publicly traded company in Spain. As such, I need to draw your attention to these forward-looking statements. We are basically a company developing epigenetic drugs for the fields of CNS and oncology. We have right now 2 programs in phase 2 with well-defined registrational pathways, Iadademstat in oncology and Bafilostat in CNS. Today, for the sake of time, I'm going to be focused mainly on our CNS program in Bafilostat. The company has a very experienced management team with proven track of being able to develop drug development till approval. Our chief medical officer and the rest of the medical team is based in the US, in Boston, with the exception of Dr. Ropaki, who is our chief medical officer for CNS, who is based in San Diego. Our 2 programs are basically aiming the same target, although they are different molecules and having different IP protection. Both are aiming to interfere with LSD1. LSD1 is an epigenetic target and it's a specific histone dimethylase, which is basically involved in different physiological and pathological processes. LSD1 inhibition has been proposed as a therapeutic target approach in viral diseases, inflammatory diseases, CNS disorders, and in cancer. In oncology, we know that there is an explicit, well-defined mechanism of action. There is a clear class validation, as we can see for the industry activity. And we, as Oryzon, we have recently signed an agreement with CRADA, with the NCI and NIH. In CNS, LSD1 is playing a very important role during development and after birth. We know that LSD1 inhibition can rescue different phenotypes in different genetically defined neurodevelopmental syndromes. And there is right now ample evidence of neurological benefits in the different animal and disease models. In this regard, Oryzon is enjoying a unique competitive position because we are the only company developing an LSD1 in clinic. As you can see on this slide, our pipeline is rich and is having different clinical trials for the 2 main programs in CNS and in oncology. But what you see on the red squares are basically the main investment thesis in the short term. Basically, in CNS, our clinical trial in phase 2B in borderline personality disorder, Cortico, which has the recruitment completed, and in AML, our ongoing trial, FRIDA, in refractory relapse FLT3 mutant positive patients. Let's go on the CNS stage. So we know that the mechanism of action by which LSD1 inhibition acts in the brain cells is basically by inducing the expression of genes involved in neuronal plasticity and down-regulating neuroinflammation. We also know that it modulates the glutamatergic signal. And also LSD1 inhibition is able to modulate the response to environmental stress and improving the aggressivity dysfunction and sociability dysfunction. What you see on these cartoons are different experiments done by us and by independent researchers in which you can see that LSD1 inhibition is perfectly able to restore primary axon branch. It's able to restore the synaptic response to the striatum in autistic models. We have shown that we are able to reduce aggression. I have shown that we are able to restore social interest with the 3-chamber test. When we were starting clinical development of Bafilostat, our LSD1 brain penetrant inhibitor, we proceed with a first-in-man, which was a placebo-controlled phase 1 trial with 110 healthy volunteers. We were able to demonstrate that the drug was safe and well tolerated, that it was highly brain penetrant, and we have a very good pharmacology, allowing a single pill per day. We also saw on this trial and in the subsequent trials on which we have treated more than 430 patients already, that we don't have any kind of undesirable side effects. So we don't have weight gain, we don't produce sedation or somnolence, we don't produce loss of sexual appetite, we don't produce extrapyramidal signs, and we don't have hematological impact. So the drug is not having any kind of CNS, heart, respiratory, renal, liver, or GI toxicity. We also were able to determine the recommended phase 2 dose in an exploratory trial that we did in Alzheimer's with 150 patients. As you can see on the graph on the right, there is a target engagement measurement that we do by a quantitative method. And at 2 different doses, we see different quantitative target engagement. As the tolerability, the compared tolerability between the 2 doses was similar, we decided to go for the higher target engagement doses at 1.2 milligram in the subsequent clinical trials. We wanted to see if in humans we were able to reproduce the reduction of aggression and the increase of sociability that we have seen in different animal models. For that, we did an exploratory phase 2A trial called Reimagine, which was a basket trial. In this trial, we were recruiting 30 patients from BPD, ADHD, and autism with significant degree of agitation and aggression. We were treating those patients for 1.2 milligrams per day of bafidenstat during 8 weeks. As you can see on the right panel, by all the different scales of aggressivity, we were able to see a clear reduction in the overall trial and in the individual cohort. Not only that, but we were also deciding to move forward on the borderline personality disorder. So the reason for that is that autism is a very heterogeneous disease, and the competitive space on ADHD was too crowded. Compared to that, borderline personality disorder shows a very limited competition. And it's a very prevalent and impairing disease. We have 9 million patients in the US and Europe. These patients are unstable and having extreme interpersonal relationships. They are agitated, aggressive, very frequently self-aggressive. So there is a high unmet medical need, and there is no approved drugs for this indication. So we are the only ones having shown so far improvement on BPD patients that we have seen in Reimagine, and we have also ample evidence on preclinical models. As I said, the competition here is very low. We are the only company running now a phase 2B trial, and the commercial opportunity here is enormous. So we expect that the peak sales for bafidenstat could exceed the $3 billion per year. With that in mind, we started Portico. Portico is an adaptive, randomized, double-blind phase 2B trial with bafidenstat in borderline personality disorder patients. It's a clinical trial who has recruited 210 patients in the US and Europe. The recruitment has finalized last July. This trial has 2 different primary independent endpoints, meaning that if we get a statistical signal in one of the primary endpoints, we can declare a success on the trial. The first one is the overall clinical improvement measured as the BPD checklist scale. And the second one is the improvement in aggression measured by the evaluation of the difference of agitation aggression CGI scale from baseline to a specific week. During this trial, we have done a number of safety analyses by an independent data monitoring committee, and we have seen that the drug is safe and well tolerated in keeping with the previous safety package. We also ran a pre-specified interim analysis once the first 90 patients were completed in the treatment. And this pre-specified interim analysis determined that the trial was not futile and that we didn't need to increase the number of patients. As I said, we got the last patient recruited in July. So we expect the last patient out in November, and we hope that we will be able to get them to release the top line data of this trial in the first quarter, on the first compasses of the first quarter next year. So this will probably suppose a transformational change for this company, because we are talking here about a unique competitive position in a very large CNS indication with little or no risk of generification. The asset is also being explored in other indications in the space, particularly in schizophrenia patients, where we have right now Evolution. Evolution is a double-blind phase 2B trial that tries to determine the improvement on negative symptoms and the improvement on cognitive impairment associated symptoms. The trial is being run so far in Europe, but we hope to be able to extend this to other geographies soon. This is a space where there's an enormous medical need. We need to remain that patients with negative symptoms and cognitive impairment-associated symptoms have no approved treatment so far. This is a crowded space on the primary symptoms, but there is a real opportunity for negative and cognitive associated impairment. On top of that, and very briefly, I will also refer to our program in oncology. So this is the second molecule of the company called yadademstat, also an LSD1 inhibitor. We and others in the space have determined how LSD1 is required for cancer stem cell survival, and which are the mechanism, the precise mechanism by which LSD1 inhibition disrupts the oncogenic program. We were recently, last December, presenting in New Orleans at the ASH conference, the final data of phase 2A trial that we ran in elderly and fit AML patients in combination with azacitidine. The primary endpoint of this trial was dose finding, safety, and tolerability, and the secondary endpoint, the usual efficacy parameter. So we were shortlisted at one of the 25 most relevant AML communications to be considered for the 2023 highlight of us, and that was because the combination was safe and effective. Response were rapid, deep, and durable. We were getting 80% of responses considering PR, CRIs, and CRs, and 2/3 of them were good quality responses, CR, CRIs, and we were seeing basically also very interesting responses in patients with a diverse array of AML mutation and response in patients with respond poorly to the current Ben-Aza standard of care, particularly p53 mutant patients and myelomonocytic leukemia. With all this, the company has positioned his molecule in refractory relapse AML patients having the FLT3 mutation, which is the most common mutation in the space and where there is still a significant unmet medical need, despite that there has been a recent approval on this particular indication. These patients are right now having only incremental benefit, and we believe that due to the fact that gilteritinib, which is the approved drug for those patients, has an enormous synergism with yadademstat, we can produce a significant benefit, increasing the response to these patients. So this is FRIDA, basically where primary objective is evaluating the safety and tolerability and determining the recommended phase 2 dose of this combination, and the secondary objective to evaluate the efficacy of the combination by CR rate, the duration of response, and MRDs. We expect to recruit up to 50 patients. The study is being conducted only in the US, is currently recruiting, and is being led by the Massachusetts General Hospital. We have also a program in neuroendocrine tumors with there is abundant evidence that LSD1 inhibition can produce particularly benefit on ASCL1 subtypes of small cell lung cancer, but also by boosting the ICIs, the immune response, and synergizing with immunocheckpoint inhibitors. We are running right now phase 2 a study of yadademstat in combination with paclitaxel in platinum-resistant refractory relapse small cell lung cancer and extrapulmonary high-grade neuroendocrine carcinomas in collaboration with the Fox Chase Cancer Center. This is a trial which is recruiting, and we hope to be able to communicate the first levels of responses during 2024. Just to wrap up, I mean, Oryzon is a unique dual epigenetic proposition in CNS and in oncology, and we are having multiple inflection points coming soon, particularly top-line readout in borderline personality disorder, our trial Portico, which we expect to get the data in January. And during 2024, we are also expecting to be able to read out or to be able to communicate the progressive readouts of our clinical trial in second-line AML patients having the FLT3 mutation. With all this, I would be very happy to participate in this conference and to take any kind of questions that you might have during my stay in New York. Thank you very much.